So your specimen will be right at the base. You apply a voltage, and then what happens? Electrons actually move to your specimen. Hello, somebody has their, hello? Can you turn your volume down? If you please check your volume, because there's feedback here, and then people are hearing your conversations and things like that. All right, you can actually turn it up before they ask a question, no problem at all. All right, guys, so again, here's my specimen at the base of the microscope, and this is electron microscope. You add a voltage to this microscope, this gun shoots off electrons. Those objective lenses, which are magnets actually, they direct those electrons to where your specimen's at. Electrons hit and it's deflected. There's a detector that picks up those signals and it converts it to an image here. Now notice this is scanning electron microscopy. You scan the surface, like you see right over here. This is a starch grain. That's what it looks like. This is a lady beetle foot pad. Take a look at this. You can't see this with the light microscope we have in the laboratory. You can't resolve that image, all right? There's a bacterial cell. You can see surface areas here. So again, scanning electron microscope scans surfaces of structures. And again, I'll show you some pretty cool images back here. Now, the next technique is called transmission electron microscopy. The setup is basically the same. So we have an electron gun, we have a specimen, but it's not at the base. It's somewhere in the middle of this little tube back here. You add a voltage to the equipment, electrons fly through your sample. This is what you would see. You see right through the sample. This is a mitochondrion, by the way. I'm not asking you to identify the structure, but you can see a very different image. So let me go back to the scanning. You scan surface structure, right? And this, so that's scanning electron microscopy. When you look at transmission electron microscopy, those electrons are moving all the way through your sample. Now you can see internal structures. All right, that's a mitochondrion, very fine. Notice this right over here. Here's the limit of resolution. It's in the angstrom range. This is 1/10 billionth of a meter. So again, it's highly resolved. All right, so hopefully you get a sense of microscopy. And by the way, I'm running through this kind of fast, but you're going to see bright field microscopy again in the laboratory. That's what we're doing this week. And again, we talked about that during the lab last week. All right, do not worry about this slide. All right, this is a calculation to show you the reason why different microscopes have different resolving power is because of the wavelength. All right, and a few factors, and this is called the refractive index sin alpha. That's something else. This slide is not for you, so don't waste your time studying that slide. All right, this table kind of puts things together for you. And again, all I want you to get about with to understand about microscopy is some general concepts. Resolution is really important. That's the most important feature for microscope. It's the ability to distinguish two objects as distinct objects. Dark field, only example I want you to know, we can use that technique to actually see the organism that causes syphilis. You don't have to tell me what that name is yet. We're going to talk about that later on in chapter. Right? Okay. Now this week, here's what we're going to be doing. And again, this was alluded to last week in the laboratory. And here's the big thing. To see bacteria nicely, you have to stain them. That's important, all right? If you're going to take some saliva, put it on a slide, look at this through the microscope, you won't see things very well. But if you add a stain to that, the bacteria stains, and now you can see morphology, right? So I just want to talk about three techniques here. Simple stain, negative stain, and a gram stain. If you recall, the most important stain in microbiology would be a gram stain, all right? Let's go through each of these, and then we're done with this lecture. Now, a simple stain, it's a basic dye. So I'd like you to know that little fact. A simple stain is a basic dye. A basic dye has a positive charge. I'm going to lay out what I want you to know. Basic dyes have positive charges. Now, here's the cool thing. If bacteria have cell walls, and not all of them have cell walls, by the way, but imagine there's a bacteria with a cell wall. That cell wall has a net negative charge on it. So the basic dye is positively charged. The bacterial cell wall is negatively charged. You guys know this, opposites attract. So what happens if you add the stain to the bacterial cell, it's going to stain with whatever color that dye is. So when you look at it through the microscope, you're going to see that color. And again, we're going to see that this week here during the laboratory. So some examples would be crystal violet, methylene blue, and safranin. These are stains that are basic dyes. They have a positive charge. They will bind to the bacterial cell walls that negatively charged. And again, safranin, for example, is a red stain that will show up as red. Crystal violet or methylene blue, they're blue or purplish. So if you were to stain the bacterial cells with this stain, that's the color that you would see. All right? Now, a negative stain, on the other hand, is an acidic dye. Acidic dyes have negative charges, right? Look at this. 3 of them I'd like you to know: nigrosin, we have India ink, and we have Congo red. So these here are dyes, but they're acidic dyes. They have negative charges to them. Now, the bacterial cell, of course, is negatively charged. So imagine if you were to do this. You add the dye to the bacterial cells. Well, they're going to repel each other, right? Because of the 2 negative charges. So the background stains, but you notice the bacteria doesn't stain. Unlike what we saw before, the bacteria stains here. In this case, the bacteria doesn't stain, but the background stains. Why? It's because acidic dyes have a negative charge, bacterial cell walls have a negative charge, and they repel each other, so the cell wall is not being stained, but the background has to stain. Now, what I'm trying to get to is this one here. The most important stain in microbiology, it's a gram stain. So I just want to go through the basic protocols. You guys will be doing this stain next week. All right, not this week, but it's a very important stain. And you're going to see why in a second. All right, how do we do this stain? Well, yes, you make a smear, a bacterial smear, you put the bacteria on a slide, dry that slide, and then you do a very specific process here. It's a very specific protocol. It has to be done in order, all right? And again, you guys will be doing this in a week from now. This stain is pretty old, actually. This goes back to 1882. It was invented by a guy called Christian Graham, hence the name Graham stains. But it's pretty cool. Here's what it does. It separates bacteria into 2 broad categories, all right? So let's go to the technique back here. And again, we're just laying it out. You guys would actually do this procedure later on. You have 4 different steps. And although I'm not going to repeat myself, realize this. Between each step, I'm going to wash with water. Just rinse out with water. So you make your smear. You add the first stain. The first stain is crystal violet. Well, that's a purple color. So this is what it looks like if you look at the bacteria through the microscope. The second step is adding iodine. Iodine is called a chemical mordant. We're going to talk about that again in the laboratory, but it intensifies the color. The third step is an alcohol wash. So alcohol, here's what alcohol wants to do. It wants to strip that bacterial cell of the stain. You want to remove that stain. So if you have a very tight binding of the stain to the bacterial cell, alcohol has a hard time washing it out. But notice this, for the gram positive, that's a pretty tight binding. That stain is pretty much bound pretty tightly to the bacterial cell. Like a gram negative back here, not so tight. So alcohol washes that stain away. So this is now colorless. Then we add a counter stain. A counter stain simply does this. It actually stains what's not stained. So by here, this is already stained. I don't have to worry about that anymore. You remove the stain from the gram negative one. When you add safranin, this will now appear red. So I have 2 different colors. Bacteria that are gram positive, they would stain purple or blue. Bacteria that are gram negative, they stain orange, red, or pink. So here's what you've done by a simple staining technique. You've just separated bacteria into 2 broad categories. I'm not looking at their names now. I'm just talking about their staining characteristics. One would be blue, and another would be red, pink, or orange, all right? That's really important because sometimes even just having a stain can direct you to treat a patient with appropriate antibiotics. And again, we're going to talk about this in the lab later on. So let me show you some pictures of what these things look like. So keep in mind, I want to re-emphasize, we're going to go through all of this stuff in the laboratory with the acidic dyes and basic dyes. We're going to do that this week. So that's going to reinforce what I'm saying here. And the next week, we're going to do a gram stain. You guys will know all the details about this, all right? So gram-positive bacteria, if you remember, let me go back to this slide. Notice this, they're purple or blue. Gram-negatives, orange, red, or pink. So after, in this case, you did the gram stain, and this is what you see. Well, I see purple, I see blue. You know that's a gram-positive, those are gram-positive organisms. You have cocci, which are round, rods, they look like this, but those are gram-positive, all right? And again, If you went to lab last week, I have the slides there for you. I listed the most important organisms that you should know, at least for the lab, that stains gram-positives and gram-negatives. Again, let me go back so we're not lost here, right? Here's what a gram-negative organism would look like. It's going to be orange, red, or pink. Take a look at this. Right there. That's what we see. It's not blue, it's not purple. So these are gram-negative organisms. Here's E. coli. It's a gram-negative rod. Here's a spirillum minor. This is also, that's a spirochete, but it's a gram-negative organism. All right, so why is this so significant? Every time we go through lectures like these, I want to go back to the clinical picture. I want to talk about disease, right? Now, let me give you two examples. The first one is quite obvious. In fact, when you learn about gonorrhea, for example, you're going to say, wow, imagine you're a nurse and you're in the ER, somebody comes in with symptoms like this. You already know what that disease is. And it's really simple. So let's go through it, all right? There's a male patient, has gonorrhea. You see this pretty thick, purulent discharge. That has pus in there, meaning there's white blood cells in there. Here's a cervical opening. You can see the same type of discharge being exuded from the cervix. It's pretty thick, purulent, thick, all right? Yellowish, whitish. If you were to do a gram stain, that's what we're talking about. This is what you would see. You see white blood cells and you see gram-negative cocci. That is the organism that causes gonorrhea. It's called Neisseria gonorrhoeae. So gram stain helps you in literally diagnosing this disease. Here's a culture. A culture means to grow. You're going to grow the organism. This is what Neisseria gonorrhoeae looks like when you grow it in the laboratory. But a gram stain takes 5 minutes to do. You take that specimen, you take a swab, collect the specimen, send it up to the microbiology laboratory. The first thing they do is a gram stain. You do a gram stain, you get a result back. It's gram-negative cocci. These are round organisms. Came from what you observed in that patient, right? You know most likely this would be Neisseria gonorrhoeae. All right, so that's a good example. I'm not going to talk about acid-fast stains here, but I do want to introduce it. So here's what we talked about staining. Simple stains are basic dyes, they're positively charged. Negative stains are acidic dyes, they're negatively charged. Gram stains, it's a pretty cool staining technique. It allows us to separate organisms into two categories. One that's gram-positives, typically blue or purple, and then the gram-negatives, which are orange, pink, or red, right? There's another stain I'm going to mention, and I want you to tuck it in the back of your mind. If everything goes well, we probably will be doing this in the laboratory later on too. It's called an acid-fast stain. So when you hear that word, acid-fast stain, I want you to think Mycobacterium tuberculosis, TB, and leprosy. Those diseases are caused by organisms in the same genera. They're mycobacterial species. So again, we use this staining technique to help us see or visualize organisms that cause TB or organisms that cause leprosy. All right, so don't forget it, an acid-fast stain. Specific, not for your typical bacteria, but specific for Mycobacterium, like Mycobacterium tuberculosis and Mycobacterium leprae. And again, I'm not asking you to memorize genus species name yet, so I want to be sure that that's clear for the lecture, right? When we get to diseases later on, that's when I'm going to introduce their names to you. For the laboratories, I do want you to know that. So if you are paying attention to me, because the laboratories, of course, is not part of this class, it's a separate lab, but we see things in common, right? So, practicum, for example, we'd expect you to know names, but all those things are listed in that PowerPoint that we gave to you 2 weeks ago. All right. And again, this is a person, just pay attention to this slide. This is a person with TB. This is the sputum from that person. You notice it's bloody, right? That's not unusual. When you have a person with TB, that disease used to be called consumption. You start destroying teeth. Go along. When they cough up sputum, it's not unusual to see blood on there. You take that specimen, put it on a slide, and you do the acid-fast stain. You see these red organisms, that suggests that patient has TB. All right, so that's a different staining technique. And again, it's specific for tuberculosis. All right, guys, so we're done with this lecture. And do you guys have any questions specifically on this? And now I want to continue with chapter 4 after this one. So let me ask you guys, are we good with chapter 3? Again, the staining techniques, but you had an in into this because we talked about this last week in the lab.